So, good morning. We'll continue from yesterday's class. I started to introduce the erasers, that's histone demethylases, which is very important players to generate the dynamic epigenetic modifications on these stones, which obviously regulates the gene expressions. Um, this also I introduced to generate the dynamic histone code, which I'll talk about very briefly end of this class. Uh, the writers so far we discussed is essential, and as well as the erasers, which I'm going to talk today. There are two major proteins which play a role in the demethylation of these stones. One is an LSD, that is a lysine specific demethylase, which specifically demethylates on the gene activation mark. That means it is the repressor proteins because it removes the activation mark. So, by demethylation of the activation mark, this makes sure this converts active genes to the inactive genes. That is what represented here when it demethylates the lysine 4, which is an only well known gene activation mark in connection with the methylation, which produces the inactive chromatins. The other family, that is a Jumonji family, it's the largest family, rest all the positions, K9, 36, 27, H4, K20, in all the positions are demethylated by this family of proteins called Jumonji family of proteins. In this case, where it can demethylate the gene repressive modifications, histone 3 lysine 9, and then when it demethylates, then it becomes part of the gene activation complex. It's an opposite to the particular mark. So, it converts inactive mark to the active mark. So, that means you can imagine, so these proteins, it will always coordinate with the corresponding repressor or activator. So, in this case LSD likely is going to coordinate with the repressive proteins. Repressive proteins, histone 3 lysine kainin methyl transferases, K27 acetylase, sorry, methyl transferases, 36. Okay, so in all these methyl transferases, this can form a complex and then work together to have a successful inactivations. Similarly, the Jumonji also works along with the gene activation complexes. It is had proteins or MLL, which is protein which methylates at histone 3 lysine 4, right. So, this is how it can work for a successful gene inactivation or gene activations. So, coming to this first family, the lysine specific demethylase 1 LSD proteins, it is histone demethylases. It belongs to the amine oxidase family. It primarily demethylates mono and dimethylation of the histone 3 lysine 4, also trimethylations. But on biochemical studies at in vitro level, it efficiently demethylates di and mono compared to the tri, which tells that in these cells it is possible it needs another complex proteins for efficient demethylation of the Me3 to Me1, sorry, Me2 followed by Me1, then Me0. So, to this pathway in a cell, then it needs maybe complex proteins, but in vitro level it efficiently methylates mono and di compared to the trimethylations. You can see this is a domain architecture. The oxidase domain is the catalytic domain for the demethylase. So, there are studies have indicated, as I said, the complex determines the specificity because the few studies very recently has been shown LSD, it is not the obligate H3K4 demethylases. Yes, majority studies confirm it is demethylase, I mean H3K4 demethylases, but the few studies are shown that depends on the complex in which associated. The LSD, this protein can also demethylate histone 3 lysine 9 Me3. So, basically demethylation only. That means LSD can also function as a gene activator protein because it can demethylate the repressive epigenetic modification. But of course, the major discoveries or major studies are reported specific to the H3K4. That is why it is considered as a part of the gene repressive complexes. Good. The Jumonji is the second largest family. This has these the oxygenase domain. There are more than 27 family, 27 proteins are in this family. The most of the proteins characterized under this Jumonji family are known to demethylate primarily H3K9 or H3K27, H3K36, and H4K20. Sorry. So that means Jumonji primarily seems to be demethylases for the repressive epigenetic modification. That means Jumonji are gene activator proteins. The name was they given because it is a Japanese name. When they were trying to do the knockouts, gene deletions, and then gene trap studies in the mouse model, they found when they delete this particular family protein, one of the protein, and they found the neuronal cells becomes something like crescent or cruciform shape. So basically Jumonji meanings is a cruciform in a Japanese term. So when because of its knockout phenotypes in neuronal cell line, and then they given the name. Later they found it can efficiently demethylate the histone proteins. So that is what the name was initially was given to these proteins. Yes, it has a conserved motifs, and it can also the various regulatory domains, because the demethylase also can carry the reader proteins, so reader domains. The histone epigenetic mark specific reader domain also can present in the demethylases along with the Jumonji domains, that is the catalytic domains. See the how do you estimate the demethylation efficacy? There is a two assays. One assay, you have a substrates, right? Histone peptides or histone protein which is methylated, then add a proteins which is a pituitary protein suspected to have a demethylase activity. It can demethylate. Then what is the output? How do you read out? Is you can do Western blot. Western blot for a modification specific, right? The K4 means K4 methylation antibody you can use. K9 means K9 methylation antibody you can use. The other strategy, this is the most friendly, user friendly approach. Quickly you can do the assays, but it's not highly sensitive, because Western blot can always bring some kind of non-specific signal as well. So the best strategy is again you have to think about using MALDI MS. Sorry, MALDI. So which can tell you the presence or absence quantitative information about the methylation mark on them. So these are two strategies always people they use to discover the demethylation activity. But as of now, majorly used Western blot based strategy. That's what has been shown here. With LSD1 specific activity was shown, and after that they see that the loss of signal for the H3K4 MA3, which is not the case with the K9. K9 signal can be picked up, but it is, you see, when they incubate with the LSD1, they see only the K4 modifications decreased. I mean, it's demethylated. And most of the studies are confirmed it is a part of these several repressor complexes that clearly suggested it is a gene repressor protein, because it efficiently demethylate the gene activation mark, right? So LSD means you should keep in mind it is the H3K4 MA3 demethylases, right? So how does it do? How these sequence? In case of DNA methylation, you can recollect it is a sequential reactions. That makes sense also. Naturally, that is how it evolved, because it produces intermediate mark that is also has some function in the early embryonic stage development in DNA demethylations. That means cytosine methylation to hydroxymethyl cytosines, intermediate mark. So it doesn't immediately go away. It stays and then it has some functions, and then goes for sequential conversions. Whereas the lysine specific demethylases, in this LSD1, both the family uses different cofactors. LSD uses FAD as a cofactor basically. Okay, LSD is a family of protein which use primarily FAD as a cofactor. Then it releases the formaldehyde as a byproduct, and then it produces the uh, lysine. Okay, histone 3 lysine 4, that's it. So Me0 mark. So for Me3 it's simple, it is oxidation reactions or hydroxylations by adding this the oxidase enzyme, and which is a primarily FAD dependent. The byproduct is formaldehyde. The single direct conversions. Whereas these the Jumonji family, which is demethylases, it follows similar to this almost adenine demethylase. If you recollect adenine demethylase, I said tryptoprotein, FTO, right? Fat mass obesity protein, and then second LKBH5 protein. Okay, so this is LKBH protein is the one. So you see both the protein, Jumonji proteins as well as this protein, which acts, Jumonji acts on the histone proteins, and then LKBH acts on the mRNA. So mRNA demethylase. But mechanistically, the chemical reaction on these demethylation similar, doesn't follow the again cytosine methylation of DNA. So look at here adenine, and then where it needs alpha ketoglutarate as a cofactor, and look at here methyl lysine, it needs alpha ketoglutarate as a cofactor. Now both the here LKBH5 enzyme and then histone demethylase, in this case Jumonji 5. This also needs iron, ferric iron, this also need iron. So both almost follow same reactions, but the substrate is different, and both releases formaldehyde as a byproduct, including LSD1 also releases formaldehyde, but that needs FAD as a cofactor, and this needs alpha ketoglutarate as a cofactor. You can imagine how the metabolic products, intermediate product, how it can influence the epigenetic outcome. You can underline, you can think about it, how food habit can influence the epigenetics, because it can influence your metabolic byproducts. Obviously this is going to influence your activity of the epigenetic protein. In this case, one example is the level of alpha ketoglutarate, availability of the alpha ketoglutarate is essential for a successful activity of histone demethylases. If there is an issue with this availability of this alpha ketoglutarate, which is an important cofactor, then you have deficiencies or inappropriate activity of histone demethylases, that will influence your obviously histone code generations, right? Both formaldehyde is a byproduct, and then it produces the adenine, in this case lysine. So this is how the histone demethylation works, and almost similar to this adenine demethylations or mRNA demethylations. So out of the initial days they identified these histone lysine demethylations. It's a classic. This is a classical protein purification strategy from this whole cell lysate. The nuclear extract, because it's a nuclear protein expected, because it's demethylates histone proteins, and the nuclear extract protein subjected to this various level of purifications in different columns, and then you see the fractionation, the purified fractionation protein. It's a crude extract, and this they subjected for the demethylase activity. In this case, what they do, usual strategy, they collect different fraction 1, fraction 2, fraction 3, and each fraction they conduct for activity, and then they'll know, okay, the fraction which carries the good activity, they'll look at it, okay, this is the fraction carries the demethylase. This is how before these when they started using the classical cloning protein production approaches, they use the crude lysate and the fraction based assay they conduct. Yeah, they used to identify the histone demethylase also, they followed the similar strategy. Yeah, so there are a lot of protein, as I said. The Jumonji protein is conserved on across the eukaryotic organisms and the model organisms. The common is Jumonji domain, the catalytic domain, which all follow the same mechanisms. It needs alpha ketoglutarate, and then it produces by the formaldehyde as a byproduct, and converts methylated lysine to unmethylated lysines. Similarly, methylated adenine to unmethylated adenine. Right. So, to have a successful epigenetic indexing, in this case, histone code, I will come to the point very briefly later. On the chromatin domains, the methylations and then demethylations are required for the balanced gene expression. In this case, balanced coordination of methyl transferases, demethylase activity is essential to generate the histone code. You see here, this is the protein which is homolog of LSD in the Drosophila. LSD homolog. LSD1 is in a human mouse. Its homolog in the Drosophila is ZUFR 33. Okay, ZUFR 39 is a methyl transferase. 33 is the demethylases. So, it demethylates on these lysine 4-Me2 and Me3 as expected, and then what does it do? It facilitates the deposition of S3 and Me3. You see, it not only demethylates, it also helps to recruit the methyl transferases. So, this is how it works. Similar to these kainine demethylation also, it demethylates kainine and helps to recruit the K4 methyl transferases. So, this is well established for these various demethylation sites. In this particular positions, you have a K4 and Me3, which is a gene activation mark. When the particular gene need to be converted to the repressive positions, what happens? The LSD1 comes and then demethylates. In this case, LSD use FAD as a cofactor, and then it releases, it converts Me3 to Me0, and in addition, it brings the ZUF3H1. It is an enzyme which methylates at K9 Me3 mark. So, this is demethylation to the methylation of this, basically activation to repressions. Right. So, it also, it's various studies are confirmed. One example I tell you, the one of the studies have identified the Jumonji protein. I said it's mostly it acts on the demethylation at histone 3 lysine 9, and also LSD. I said at some extent it can act on the K9. So, one study has shown that in the presence of the protein, this androgen receptor protein, when it bound to the LSD1, its specificity changes. So, the LSD1 primarily demethylates at these K4 positions, but in a complex with the androgen receptor, it can efficiently demethylate to the K9. That means, in complex with the androgen receptor, the LSD1 usually it is a repressive proteins, but now it becomes an activator protein. So, you see how the epigenetic proteins are dynamic in response to the in association with the complexes. A similar, it has been shown with the Jumonji as well. So, not with the K4, but different positions. Right. See, Jumonji in connection with the K9 and K36, I briefly highlight. I am not going to talk about the whole Jumonji family. The 4 protein, I will just show you for your example. Jumonji D2 is a very important protein. Okay. Jumonji D2. In there are 4 protein A, B, C and D. You can see here. C, A, B and D. So, out of that, this A is a protein which has a dual demethylation activity. Dual means what I mean, it can demethylate both repressively K9 and K36. The same protein can efficiently demethylate both the positions. It's very unique. Rest all the proteins, mostly it is demethylates at histone 3 lysine 9 Me3. So, the among these major family of Jumonji, most of the proteins are known to demethylate K9 except the D2A which efficiently demethylates in both the sites. So that's what I was trying to explain here. Uh, uh, um, the K9 demethylases and 36 methylases, the primarily for this D2A, at some extent to the C as well, B and then D. So these are just for information. Keep in mind the background information. Jumonji family is the demethylases for K9. Of that, there is one protein or maybe two protein. D2A function is both K9 and 36. So this is as a readout what I explained when I started to introduce. You can use MS, multi-MS to see the demethylation on the peptides or histone proteins or the Western blot approach using antibody-based strategy. So you take histone proteins and incubate with this demethylases. In this case, Jumonji D2A, you see here, and which can efficiently demethylate K9 and then 36, but not on the K4 and then not Me2 as well, okay, and not Me2 or K9. There is an important control is missing. So what is the control? You can think about it. Housekeeping genes, right? But in these connections, what is the control? You can think about it. How you make sure, forget about this, okay? How you make sure this loading is perfect or not? What is the control? You should think about it. Histone 3 protein, right? Here modification specific antibody. So you should load another control histone 3 where I have my equal histone proteins here, both the case, but only modification changes happen, right? So I see the decreased K9 and then K36 trimethylation mark, but my histone levels are similar. So this is how you will make logical conclusions, because the antibody can little bit cross react as well. Because see, imagine you have a antibody having small chemical modifications, trimethylase, 3 methyl groups, that becomes an antigen, an epitope, small epitope, right? So the antibody cannot, unless until you have very specific monoclonal antibody, it cannot have a strong differences. So that is why you have to be very careful when you are reading or conducting these kind of assays. But if you have this control, it makes sense. That is your solid identification. Otherwise, you have to think about this kind of experiment, which is expensive or it depends on the and equipments. Another simplest assays, which is not a very strong assays, but still one can always imagine. Okay, whatever the two assays which I explained to study the demethylation is the in vitro assays, right? In the cellular level, one can always think about it. When you overexpress the protein, Jumonji 2 protein, you see the decreased K9 and 36 trimethylation. You see, again, this is antibody based strategy. If you look at very carefully, um, um, the green is demethylase, this place, okay, and then the red is 36 antibody, Me3. Wherever you have the green dots, that place you don't have much of this place, the red color staining, that says that particular cell is demethylated the 36 positions. That is the results. Similar to this here, you can see here. No, K4 actually it's not demethylated. That will say the K4 antibody is staining here, same to the cell. If you look at very carefully the K9, this wherever you have the green cell, right? Green cell is producing the demethylases, and you don't have the red color staining here. Got it? So this is immunofluorescence staining. That particular cell lost or demethylated, because other cell you have the signal. You see, other cell there is no demethylases, because in the transfection you don't have any control. Every cell get transfected. Wherever you have the transfection, that cell produces the protein and then it demethylated. That is the logic. You have, okay, it's a very beautiful experiment. They see increased expression of these Jumonji D2A protein in this cell, no signal, but adjacent cell has a signal, which tells that okay, that particular cell lost. So this is the way also they confirmed this Jumonji D2A can demethylate K9 and then 36. Okay. Okay. So I explained about LSD, right, and then Jumonji. Both I explained the mechanisms. And Jumanji, mostly it is a repressive specific demethylase, repressive MOC specific demethylase. That means it's activator proteins. And I'm going to explain very briefly for the 27 demethylase. There's only one protein. I don't know how many of you recollect when I talked about polycomb repressive complexes. I briefly talked about this demethylases. So there was a protein which I explained, UTX. Okay, it is also carrying the Jumanji domains. Okay, this also carries, but very specific protein which specifically demethylates at histone 3 lysine 27. You see the beauty of 27 positions. Methyl, the writer also only one protein, that is enhancer Z homolog 2. Sorry, enhancer Z homolog 2, which is a writer for H3K27, and then eraser also it is UTX. UTX is ubiquitously transcribed tetratricopeptide, which is present on the X chromosomes. UTX protein, which is having a Jumanji domain, which is specifically demethylates at a histone 3 lysine 27 Me3. So it is a activator complexes because it removes the repressive epigenetic modifications. UTX is an activator complex. So this is the biochemistry assays you have to keep in mind. So how do they identify? Again, all Western blot based approach. It's a crowded slides, but I'll just point out, you'll just look at those positions. So this is an assay with the UTX protein. They conducted with the recombinant, not recombinant, histones extracted from the cells. Total histone extract that has a various modifications, all the modification. With that, they conduct the demethylation assays. That means in the presence of alpha ketoglutarate, right? And you see the when they and then they separate and then they probe with the various epigenetic modifications for K9, Me2, Me3, K4, Me2, Me3, and then histone 3 lysine 27. In all the places they see signal. Okay, but whenever they probe with the Me3, histone 3 lysine 27 Me3, they see decreased signal. Right, so this is how they confirm. Okay, UTX can demethylate only 27, not on any other positions. Again, this is a extended assays. It doesn't demethylate S3, H4K20, 37, none of the positions. Almost all the major. So here in this case, 36, 79, H4K20, S3K4, S3K9, all the major histone epigenetic modification they tested, and then the recombinant UTX protein, and it demethylates only in a 27. To validate further, this is another assay they have done beautifully. They made a mutant protein on a UTX. But mutant is catalytically inactive. The key residues you can make a mutation. In this case, histidine and glutamic acid they mutated to alanine, double mutant. And you see here the wild type protein, no signal. That means it erased, it demethylated. Whereas the canine is intact here, and then the mutant protein, although they use the same concentrations, they see signal. So this is the assay you always design to conclude, yes, whatever the protein I discovered is specific to these particular demethylases. And to know either which cofactor specific, this is another beautiful biochemistry assays. You see they use ascorbate, alpha ketoglutarate, these are all for informations, and then ion. Whenever they have alpha ketoglutarate and these ion, then only they see the specific demethylase activity here. No, this case they want to say in addition to the ascorbate as well. Okay, very specific assay, and then they see loss of decreased methylation activity here. So all the biochemistry confirms the histone 3 lysine 27 specific demethylases are done by UTX proteins. Okay, this is cellular assays. So where similar to these localizations, they have done a luciferase based reporter assays. The when they knock down the UTX proteins, they see increased methylations and then decreased gene expression. This makes sense. And similarly, overexpression also they have done a similar level. And then this is another assays to show the UTX protein. its presence on the particular genes, it's a typical chip assay. So chip assay they conducted to show its protein is enriched on the OX gene. So which is makes sense because actively expressing genes where it can demethylate the histone 3 lysine 27. Okay, so also there is one more studies are shown that the UTX protein make a very stable complex with the MLL, which makes sense, right? MLL is the histone 3 lysine 4 methyl transferases, and then UTX is the demethylase for the histone 3 lysine 27. So you are converting inactive genes to the active genes. So it's not only you are removing the repressive modification, you are also bringing the activation mark. The same logic what I explained for the LSD in Drosophila, right? So in this case, it makes a strong complex with MLL1, and then this can demethylate the UTX, and then it can bring the gene activation mark. Okay, so now I explained about the UTX, which is the histone 3 lysine 27 demethylases. It is a gene activator protein. The last demethylase which I am going to explain, which is specific to the 36, which I already briefly talked about it, Jumonji D2, where A, J, M, J, D2A and then D2C. These are the two major proteins which demethylates at histone 3 lysine 36 Me3. Right. So this is the information about these 36 complex. Writers are said D2, erasers are the Jumonji D2A. What I explained, and the readers, it is a PW domain, which I already explained to you guys. See, it does also follow same mechanism, but the major protein you have to keep in mind D2A and then C. So these are the proteins which can efficiently as of now, but other protein also they reported, but at least these proteins you should keep in mind. Sorry, this year it can demethylate these positions and then convert inactive genes to the repressive genes. All right. So with that, I'm concluding the eraser proteins. Okay. So so far we have discussed in detail writer in very detail. I hope so. And then a reader protein, I talked about it, and then eraser, the today's class. You will come across this term in some of the something like DNA code. Some literature started using the histone code. So I'll explain, because sometimes you'll not understand when you read about it. But what is histone code? What does it talks about it? It talks about a nucleosomes which are carrying specific set of epigenetic modifications or gene associated nucleosome or gene nucleosome linked to the specific genes carrying a specific set of epigenetic modification that you define as a histone code. See, for example, gene activation histone code means it is always or most likely K4 and then acetylations. And repressive, it is possible K9 alone or K36 alone or K27 alone, H4, K20 or combinations. So that combination you define as a histone code. Okay, so that means each and every gene will have the specific histone code or epigenetic code in this case. So that few literature started using this kind of term. So you should not get confused what is histone code. Histone code is nothing but the nucleosome carrying a specific set of epigenetic modification. It can be an activator modification or repressive modification. In case of activator modification means you should think about it. What are the histone code you can consider or categorize as an activator, and K4 Me3, and then you can consider K9 acetylations or K14 acetylations. So these are all the histone code for activations. And similar to that, repressive also the various combinations you can consider. So K4 Me0, K9 Me3, K27, Me3. So this is one histone code for a particular gene maybe. This is maybe that gene is definitely a repressive gene. So likewise, the histone codes are recent days, or some literature started to cataloging repressive histone code or activated histone code and so on. So you should not confuse what is histone code. Histone code means what we discussed so far only, but it's a dynamic for each and every gene. So that defines the function of the particular nucleosomes. So I hope you are clear with the dynamic histone code or histone code at least, right? So let's recollect so far briefly what I explained. In total, for example, DNA epigenetic modifications, right? RNA epigenetic modifications, histone epigenetic modifications, and DNA epigenetic modification again, writer, reader, and eraser. And writer, it is DNMT1, 3A, and 3B, right? 1, 3A, and 3B. 3A and 3B are de novo enzymes. This is maintenance DNA methyltransferases. Reader protein for DNA epigenetic modifications are MBD, right, and MECP2. So methyl binding domain proteins, MECP2. For information, some of the commercial kit they sell you. I have the kit which is a methylation enrichment kit. They won't tell you what is inside. They'll give you a small column, and then they'll tell you this is a column will help you to enrich the methylated DNA. That column will carry the protein. That's it. They make a recombinant protein, they pack and give it to you with it. So they won't reveal it, but they say methylated DNA enrichment kit because it has the methyl binding domain protein in it. And eraser is TET, right? TET 1, 2, and 3. So these all are the DNA epigenetic players: writer, reader, eraser. Coming to RNA, writer, reader, and eraser. Writer is metal, right? In this case, I am talking about M6A. Met L314. 3 is the catalytic component, 14